I should actually call this the electric human, because I want to talk about electricity, and not just water, but electricity. Let me tell you just how we got started. It started with two famous scientists, one of whose picture you've just seen, but the first was Albert St. Georgi. And St. Georgi is considered to be maybe the father of modern biochemistry, and a brilliant fellow with wonderful quotes. He's highly quotable. For example, life is water dancing to the tune of solids. And he knew that water was central to all of biology. And then came Gilbert Ling, who's now in his 90s. This picture was taken long ago, and he's written 5 books since then. And there were several major contributions. One of his contributions was that the water inside the cell was not like this water here. It's actually water that is structured in some way, organized. And the idea was that next to protein surfaces, which are charged and have hydrophilic regions, the water would build up into some sort of ordered structure, and that the inside of your cells and my cells were filled with water. And most scientists today don't really think of water as being important. They think of water as being, well, as I said, just like that. However, we all know that we're 2/3 water approximately. And if you translate that 2/3 into the number of molecules, because the water molecule is so small, it turns into more than 99% of our molecules are water molecules. So they've got to be important for something. So I actually took off on this. It wasn't so long ago, about 10 years ago, starting from Gilbert Ling. And the idea that the water inside the cell was critical to any kind of cell function. And I wrote the book, and the book was originally supposed to be to translate Gilbert Ling's ideas into plain English and to try to expose them to the community. But I soon realized as I started writing that there was really more to it than what Gilbert had thought. But still, the idea of structuring of some sort of water next to surfaces was really important. Well, by some serendipity, we came upon an idea to test the idea of water structuring and how far the structure might extend from the surface. And so the idea at first was a simple one. We took a gel, in this case it was a polyvinyl alcohol gel, and we put it into a chamber, and we poured some water in. Water plus, we put particles, these tiny spheres, microspheres in, although later we used different solutes and got a similar result. And the idea was that Gilbert Ling brought up was that this kind of ordered or structured water would expel solutes, just like ice. And therefore, if you want to find out how extensive this region is, you can just see how far the region exists without solutes or without particles. So we did this experiment, and we found, this is the first time pretty much that we did it, is that these particles were expelled from the region near the interface. They kept moving and moving and moving, and then they'd stop at about 50 or 60 micrometers from the surface. They would undergo thermal motion, moving around, bouncing, but they would never enter back into the region, the clear region. They were excluded, and therefore we call this the exclusion zone, or EZ for short. Another example of it is shown here, and in this case we take a polymer, naphion, and it comes in sheets, so we just cut the sheet and place the sheet down in the bottom of the chamber and add the water and the microspheres and see what happens. And this is what happens. The exclusion zone builds up, and it builds, and this video is cut prematurely. It'll extend out to something like 300, 400, 500 micrometers. That's practically infinite by molecular standards. Huge numbers of water molecules in between. So by now, this finding has been repeated by and confirmed by many people. In fact, it was found, it was reported in the Journal of Physiology in 1970, and it was a bit embarrassing to find out after the fact that someone else had done it and got pretty much the same results. So we know that these exclusion zones exist, and the question is what's really going on? So the questions that I want to answer are, first, is this exclusion zone, this phenomenon general, or does it just apply to some polymers and a couple of gels? Does it really arise from the ordering of water? Because if it did, it would mean a huge number of molecules ordered, and this is against conventional thinking. And since creating order requires energy, where does the energy come from to create this if it really is order? And finally, what I want to spend most of the time on is, can the ordered zone help explain everyday phenomena? And by phenomena, I mean, for example, why do clouds exist as they do, and how come when you sprain your ankle, it swells very quickly? So the first question is the one of generality, and we've done a lot of experiments, and I don't want to bore you with all of them, but we've found that many substances show this exclusion zone, as you can see here, including monolayers. By monolayers, what I mean is single molecular layers. So you have a single molecular layer, and this exclusion zone builds, which means you need basically only a template. You don't need something that has thickness to it. And among the solutes that are excluded from this exclusion zone, it's not just the large particles that I've shown you, but solutes ranging down in size to approximately 100 molecular weight. And one example of that is shown in the next slide, where we take pH-sensitive dye. And now these dyes are mixtures of molecules whose size is on the order of 100 or so molecular weight. And it's cool because they show you local pH by changing color. And so this is an example of that. At the bottom is a piece of naphion, similar to what I just showed you. And all the pretty colors that you see are indicating different pHs. The main point of this slide is that the region right next to the naphion here is clear, free of dye. So it means the dyes are excluded. And as I said, the dyes are approximately 100 molecular weight. What's even more interesting than that is the color distribution. And that orange color that you see just beyond the exclusion zone means a pH of 3 or less. So a lot of protons are sitting right next to that exclusion zone, and fewer and fewer. And we'll come back to that because that's really important. So in terms of generality, many hydrophilic surfaces generate exclusion zones, and many solutes are excluded from it. Okay, so question 2 is this zone really physically different from bulk water? I've alluded to the possibility that it might be different, but I haven't shown you any evidence. Well, I'm not going to show you a lot of evidence because it will take a lot of time, but I'm just going to summarize most of the evidence and show you some of it. And a lot of the evidence is published, and the stuff that the rest that hasn't been published will be in my new book, which should be out in 2 or 3 months. So the first is that these easy water molecules are more constrained than bulk water molecules. They're also more stable, we found from infrared radiation coming from it. We found that the exclusion zone has a negative charge, and I'll show you those results. We'll come back to it since this is obviously central to the interests of everybody sitting here. We found also that this zone absorbs light in the ultraviolet at 270 nanometers. That number is important, and I'll come back to that one as well. We found that it's more viscous than bulk water. We found that the molecules in that zone are actually aligned in some way. And we found that the molecular structure is somehow different from bulk water. And finally, this is not our results, but the results in 2 Russian groups who looked at the optical properties of the exclusion zone, and they found that the refractive index is about 10% higher than that of bulk water. So there are a lot of things that are different in this zone from bulk water, so it's clearly a distinct zone. Now, going back to the negative charge that I mentioned, the way we did this experiment is we set up a chamber, and where it says inside, that's actually the inside of a gel. So you have a gel sitting there, which in this case is a polyacrylic acid gel. And outside refers to outside the gel where there's water. So we took 2 electrodes. These are microelectrodes that taper down to less than 1 micrometer. And we put one out here somewhere as a reference, and the second one is sitting here. And so we move it point by point toward the interface, and here's the distance scale. And if you're far enough away from the interface, the potential difference between here and here is 0. That's comforting. That's what you expect. But as you get closer and closer, you begin to pick up a negative electrical potential, which goes down to, minus 120 millivolts. So the region of negativity seems to correspond roughly to the position of the exclusion zone. So it looks like the exclusion zone might be negatively charged. Now, we get rid of the gel and put a piece of naphthalene there instead and repeat the experiment. The result is qualitatively similar, but quantitatively a bit different. The region of negativity extends farther out from the interface, but please remember that the exclusion zone is bigger in the case of naphthalene. So there's a correspondence between the region of negativity and the exclusion zone. It looks as though the exclusion zone is negatively charged. But that doesn't really make sense if you think about it, because what you've done is you've taken some water and you've poured the water in, and the water starts out as neutral. So how do you start with neutral water and wind up with a fairly large negatively charged zone? Well, the only reasonable way you can do that is if you have a positively charged zone somewhere else. And the question is, well, is there any evidence for a positively charged zone somewhere else? And the answer is yes. If you look at the previous slide that I showed you and realize it's turned 90 degrees for convenience, and realize that you do have a negative zone, but remember the red color. The red color indicates a huge number of protons. So it looks like you have negative here and positive here. So the two seem to be in balance. And just to check to see whether there really is charge separation, we put one electrode here, and connected them through a resistor. So if there's charge separation, you should get current flow. And indeed, that's what we found. So the current starts at a fairly high value and goes down not to 0, but to some plateau value, which extends over some time. So it really looks as though there is definitely charge separation between the exclusion zone and the region beyond. So really what we have looks like a charged battery in water. And of course you don't expect a charged battery in water, but it appears that's from the evidence that's what we found. Now, one expectation of this, which we checked, is that if you have separated positive and negative charge, you should have a force between the two, because the two want to attract each other. And we actually tested recently to check to see whether there is this kind of attraction. And we did it by setting up a lever, which you see up at the top, a ribbon-shaped lever, and we put a piece of naphthalene attached to it. And if you look at the second panel, you see what we expect to happen is that we have a negative exclusion zone here, and you have the positive charges that are out here. There should be a force between the positive charges and the negative charges that are attached to the naphthalene. The exclusion zone is practically bound to the naphthalene. So we expect the beam to deflect, and indeed that's what we saw. As you put the water in, the exclusion zone builds, the beam deflects. So it looks as though there really is, our charges genuinely separated between the exclusion zone and the region beyond. So I've just shown you the red one, that the exclusion zone has negative charge and positive beyond. So now if you want to figure out what's going on in this exclusion zone, all of these are clues. But the three clues I think that are the most important, starting from the bottom, the blue ones, is that the exclusion zone molecules are aligned, and they're fairly stable, and they're constrained. If you put those together, what fits most closely is some kind of liquid crystal. So it looks as though this region is a liquid crystalline region, as opposed to the bulk water, which is a liquid. So far, what I've shown you is that you have a liquid crystalline region of some sort. You have negative charge in this region. This region excludes solutes profoundly, unlike bulk water. And there's some hint that it's non-dipolar because of reasons that I'll go into in a moment. Not just a series, a stack of dipoles. And it may extend very far, but very far, we're talking not the 2 or 3 molecular layers that the chemistry books will tell you, but if this is right, it's 2 or 3 million molecular layers that we see. Now, it was suggested 101 years ago by Sir William Hardy that there might be a fourth phase of water. So we think of solid, liquid, and vapor, but there were many reasons long ago forgotten that led to the suggestion that there might actually be a fourth phase. So, you know, whether this is a fourth phase is up to you to decide or judge, but all of the physical and chemical properties of this zone are different from either solid or liquid. Now, what about the non-dipolar structure? Well, my hero, Gilbert Ling, the second person I showed you, suggested a stack of dipoles. But as we began to look at this, we realized that model is not adequate. The reason it's not adequate is that, first of all, is that this zone has negative charge. It has a lot of negative charge. Dipoles are neutral, so it doesn't matter how many dipoles you build up, stack, you still get neutrality. So that doesn't work. And the second reason is that this 270 nanometer absorption of light in the exclusion zone, usually that kind of absorption is not associated with dipoles, it's associated with ring-like structures. So that's a second reason to doubt the stacked dipole. Well, if you want to figure out what the structure might be, where do you start? Well, the way we started is by saying, probably the structure of this zone should have some precedent. It should be built on some structure that we already know about. So what kind of water structure do we already know about? And that's ice. Okay, so this is a picture of a model of ice. And on the left side, these are just two different views of the same structure. The red dots are oxygens. And in between the two oxygens would be a hydrogen. It's just omitted to reduce the clutter. Now, if you look on the right side, you see these blue dots between the red oxygens, and these blue dots are protons, positively charged protons. And so what you have is that the negatively charged oxygens are, you might say, glued together by these positive protons, and that's why ice is hard. So we thought maybe some variant on the ice structure might be adequate to characterize the exclusion zone. And so the first idea is, well, you know, let's just remove the protons. And if you remove the protons from a neutral structure, which is ice, take away the negatives, then you get, take away the positives, and then you get a net negative charge. And that's good because that's what we want. And also it's a non-solid. We don't want a solid. We want something that maybe has viscous gel-like properties. So it looked like a great idea, until we realized, scratching our head, that it won't work. And the reason it won't work is that if you take away the positive charges connecting two negatives, then you have two negatives juxtaposed next to each other and the structure would just fly apart. So a great idea, but unfortunately it didn't work. So then after a bit we thought, here's another idea, very similar but slightly different, which worked nicely. Take the two planes, shift one relative to the other by half of the oxygen to oxygen distance, then you get something that's very nice, because what you see is that you have the oxygen in the back plane, negative, juxtaposed next to the blue hydrogen, which is positive, so you have electrostatic sticking. So these planes then stick together nicely, but not as rigidly as in ice. So with this structure, you have a situation like this, where you have a material, it's sitting in water, and from the water molecules somehow build these easy layers that stack on one another one at a time, growing like this. And so that would be one way to account for this kind of structure. And if you look at one plane of this structure, it looks something like this. It's a bit like ice, but it's not exactly ice. However, if you count the number of oxygens and hydrogens, it's not H2O. It's actually it's H3O2. So you might say that this zone of water, it's water, but it doesn't have the same chemical formula as water. It's slightly different, like H2O2, for example, hydrogen peroxide, same molecules, but in different ratio. And it's that ratio that confers the negative charge on this zone. Now, in constructing it, I took the top plane and moved it to the right relative to the bottom one, but there was nothing sacred about moving it to the right. I could have moved it to the left, or I could have moved it 60 degrees or 120 because of symmetry. and I would have gotten pretty much the same result. So the ability to shift in each of 6 directions gives a lot of latitude. For example, you can build structures like this, helical structures. And so here you've got layer 0, the first layer shifted this way by 0 degrees, 60 degrees, 120 degrees, giving you a helix. So why is a helix important? Well, helix is important because, as you know, in biology, most of the molecules are the fibrous proteins, for example, in DNA and RNA, they're all helical. And it's known that these entities are surrounded by some kind of ordered water. So it's possible that this creates a nice interface with those molecules. So the advantages of this, these sheet-like layers that stack is that, first of all, it's not pulled from a hat. It has precedent. It has negative charge, which is what we need. The ring-like structures are more able, with delocalized electrons, to explain the 270 nanometer absorption that's observed. It's able to accommodate helical structures. And an important one for chemists, that the large sheets eliminate the Brownian instability. Now, what do I mean by that? Well, if you look at the structure on the left, which you may think of as stacks of dipoles, you know, each dipole undergoes some kind of thermal motion, and if you start stacking them, the thermal motion builds, and you can't build more than 6 or 7 or 8 or 10. Various chemists have said, therefore, this kind of structure is impossible. We're not suggesting, there are arguments both ways. We're not suggesting this structure, we're suggesting a structure that has practically infinite extent, something like this. And when you increase the mass, the Brownian excursions diminish based on the mass. So we have something here that undergoes very small Brownian motions, and each layer is effectively glued to the region next to it. So the issue that you can't build many layers because thermal motion will make it impossible vanishes. Now, so that works. Now, another one is that the tight lattice keeps the hydronium ions out. So what do I mean by that? Well, we've got charge separation here. We have an exclusion zone that has a lot of negative charge, and we have a region outside that has positive charge. And as you know, when you have negative and positive, they want to do that. That doesn't happen, or it doesn't happen very much, because we can put the electrodes in, and you can put the electrodes at time equals 5 minutes, or 1 hour, or 3 hours, and it's pretty much the same. So how is it possible that those positive charges, which are actually not protons, they're protons latched onto water molecules to give hydronium ions, which we all learned about in freshman chemistry. Those hydronium ions, they really want to get desperately to move into here, but they can't move in. They don't move in. That's the observation. And so why don't they move in? Well, they don't move in, according to this model, because this lattice structure is so tight. The hexagons themselves are small, but the hexagons are shifted relative to one another. So the real openings are extremely small. So they can't get in. They keep separated. So the battery charge remains separated. So another advantage, this tight lattice keeps the hydronium ions out. So it looks like there are a whole bunch of reasons to think that this model is reasonable, but you might ask, well, okay, it's reasonable, but give me some evidence. Does it really exist? There are actually quite a few papers demonstrating hexagonal types of water near interfaces, but most of them deal with only a few layers because of the methods that have been used. But there's one method that goes more deeply, and that's shown here. The paper published by a Harvard group in 2008, and they studied the protein ATP synthase, the subunit C of that protein. It's a very old protein. It has an interesting character to it. What happens is that when the atmosphere is dry, it forms vesicles that you see, for example, in the top slide, that enclose the water. So you have a volume of water enclosed in this either circular vesicle that you see at the top, or different geometric shapes that you see here, but these are volumetric structures. So what these investigators did was to take an electron beam and do diffraction on the water. This is a volume of water in between. And the result was interesting. It's a series of dots, and the dots mean that the water is very well structured. And the fact that you have 6 dots around here means it's a hexagonal structure. So they concluded that the water inside these capsules are ordered hexagonal sheets of water. So the answer to question 2 is the easy physically distinct from the bulk. Yes, it's distinct, and it looks like it's a layered honeycomb structure. Okay, so now I presented to you this battery that we have, and as you know, batteries usually need charging or recharging, and the question is, you know, what charges this battery? We couldn't figure this out for a few years. We scratched our heads, and finally we found out that, in fact, it was radiant energy, light. So light charges this battery. And we found out, actually, from an undergraduate student who was walking around. We had the chamber sitting here, and the undergraduate student had this light and was shining the light, and we saw that where he shined the light, the exclusion zone was building, actually rather impressively, something like you see here. Here are the microspheres, here's the exclusion zone, and this is the region that was illuminated. And you take the light away, and it comes back again. We found also, I won't show you the data, it's published, spectroscopic information. We found that the most powerful wavelength, among the wavelengths we studied from UV to infrared, about 5 micrometers, was the infrared, especially at 3 micrometers. And we could shine the infrared with a small light-emitting diode that emitted the infrared, and we could get an expansion easily of 10 times. You take it away, and it comes back again to its original. Now, where do you get infrared? Well, we're all familiar where you turn the range on, and it gets hot, and it emits infrared. But in fact, everything around us emits infrared. It's you might say, a gift from the sun or the stars, the cosmos, hitting the building and re-radiating on the walls and such. So this is essentially, this is free energy, literally free. It just comes to us. And this is the energy that goes into the water and builds the exclusion zone. So effectively, you have a situation like this. You have a material and water next to it. And this easy water, from the energy that comes in, many wavelengths, but infrared is the most important. It builds up like this, and if you were to take away the excess energy, it would come back. We also found a way of diminishing the ambient infrared energy. This took us, embarrassingly, a while to figure out, but if you take a simple Starbucks a coffee container, it insulates, right? And so what does that mean to insulate? It means it blocks the infrared from coming into your iced coffee and warming it up. And so we took a Dewar, the same sort of thing, but a professional one. We stuck the chamber with the exclusion zone inside. So this is the result. The top one is the control. You can see a standard size exclusion zone. And then we put it into the Dewar for 15 minutes, took it out and quickly looked before it built up again. And you can see that the exclusion zones on either side are smaller, roughly half the size, and then pull it out and let it sit on the bench, and they build up again back to the normal size. So it means that we can either increase the energy or decrease the energy, and it has a direct effect on the exclusion zone. So the answer to question 4 about energy is that EZ is powered by photonic energy, which orders the water and charges the water battery. Now, what about energy flow on the Earth or the solar system? So the sun hits the water and generates heat. We all know that. However, I've shown that it imparts energy for building order and separating charge. Now, if that's true, then you might ask, okay, if it's building up this potential energy, can you get the energy out after you put the energy in? And we found that you can. We found that, again, a couple of undergraduate students found if you put a tube made of some hydrophilic material, either a Nafion to start with or a gel, that you get constant flow through the tube. And the experiment is shown here. You take the tube and you quickly you fill it with water to make sure there are no air bubbles. Put it in a water bath that also contains microspheres, so you can see if there's any movement. Put it in the microscope, and the result is shown here. So you see, so you have the flow, and the flow keeps going. And it's not just the Nafion tube. It's also, we found the same thing occurring in a gel. We cut a tunnel inside a gel, a cylindrical tunnel, and you can see it here that there's an exclusion zone, and right in the center, are all the microspheres, and the video shows what happens. You've got constant flow through there. And recently, we tried to see if we add light, do we get additional flow? We found that we can actually increase the flow by up to 5 times by adding light. So this is flow that's basically free for the taking, and it's powered by light. So essentially, it means that this is doing work by driving it through, because the water has some viscosity. So if work is done, if the conservation of energy works, then the system must be absorbing energy, and I suggested to you what the energy is. It's the electromagnetic energy that is coming in to give the potential energy to drive the work. So water transduces light energy. Okay. Now, again, if you're not Rupert Sheldrake, you'd think this is weird. However. However, if you think about it, think about the plant that's on your windowsill. It's doing the same thing. It's taking light energy and transducing that light into chemical energy, which drives metabolism, bending, growth, what have you. And so I'm suggesting to you that the same thing is happening in this glass of water right here. The energy is coming in, and it's getting transduced into some kind of potential energy from which you can get a lot of interesting things, some of which I don't have time to talk about. So that leads to the equation, and I know the units don't match, but that you can't really think about water without thinking of energy. Somehow there's energy that's getting into this, and this energy, again, I don't have time to suggest to you, but some of this energy may contain information. That's another story beyond this. So I want to give you 5 examples briefly. The first example is the cloud. Okay, so the cloud, the question arises, you have water that's evaporating from all over the place. So it's not only evaporating from just under the cloud, it's also evaporating from here. And the question is, well, how come there's no cloud here or here? How come there's just one here? It appears that the water molecules are somehow evaporating, coming, you might say condensing, but what brings those water aerosol droplets together? And I'm going to suggest to you in the next few slides that you'd expect them to repel each other, but actually they come together because of the so-called like mechanism. They like each other, so they come together. So just to explain that, here's a summary of what I've shown you. If you have a charged particle or molecule in water, you have this liquid crystal, easy water around it, negatively charged, and you've got a lot of protons or hydronium ions spread all over. And this is powered by electromagnetic energy. So you won't find this in the textbooks, of course. But anyway, if we take this, just remember this, and suppose we have 2 particles now instead of 1, and suppose they're both negatively charged. Well, the reflexive response is, oh, they're both negatively charged, so therefore they'll repel each other and they'll move in this direction. However, they actually move in this direction. And this has been shown almost 100 years ago by Irving Langmuir, and then Richard Feynman in his lectures was talking about this, and Feynman in his own inimitable way called it like likes like. That is, like charges like each other, so they come closer together. In fact, you can find something similar, which comes from the tale of Genji, about warring parties fighting against one another. The only way to bring these parties together is to put something attractive in between. I'm told this is in the tale of Genji. So that's what happens here, and until recently, Feynman said like likes like because of an intermediate of unlike charges, and here are the unlike charges bringing them together. And the stability occurs when the attractive force is equal to the repulsive force between the 2 negatively charged entities. Then they're stable. And if they're stable, if you have a lot of them, then they're going to be in a situation just like this. And this is well observed by many groups over many years. It's called a colloid crystal. And it's like if you may have had yogurt this morning, probably the yogurt that you ate had the consistency that it has because of these opposite charges that keep the particles basically in balance. And they stick together because of like likes like. And I'd like to suggest to you that's what happens in the clouds, that the aerosol droplets all have negative charge, and what's holding them together are the positive charges in between, giving you like, likes, like attraction. And if they're ordered, you can get rainbows out of this. Okay, next point, sandcastles. Well, you know that you can't build sandcastles out of sand. You need sand plus water. So what's going on? How come you, with the water? So what happens is that you see the sand particles up there. Each sand particle, silicon dioxide, builds an exclusion zone right around it, negatively charged, and in between are positive charges. So you have attractive forces because the positive attracts the negative. It brings all those particles together and glues them together, so you can build sandcastles to protect us against invading flotillas. Okay, ice. Now ice, we find this all around the cosmos, and there's a paradox that I'd like to start with. I'd like to talk to you about how ice, I believe ice forms. The paradox is that usually if you want to create order, you have to put energy in. It's like cleaning your room. You know, it's easy for the room to be messy, but it takes energy to restore it to its ordered state like that. But in the case of ice, it looks like, you know, you put it in the freezer, it pulls out energy. So it seems to be the opposite. How do you get energy withdrawal? It seems to be philosophically or thermodynamically opposite what you expect. And does ice really violate the general rule? So let's look at how ice might actually form. So this is ice. You saw this before. As I showed you, the ice structure is very closely tied to the EZ structure. Just a slight difference. You just pull out the protons, remove the protons from ice, and you get the exclusion zone. So these are very close together. And the implication is, if you want to get ice, you just have to start with the exclusion zone and re-add the protons. So this is a reversible situation. It looks like the exclusion zone, being so close to the structure of ice, looks like a possible precursor of ice. In other words, the hypothesis is that in order to get to ice, you don't start with bulk water. You go from bulk water to EZ water and then to ice, because those two structures are very close together. And so it would work something like this. You start with the EZ structure, right? And then you add protons in between those two layers, and then the layers undergo a bit of shift, as you see here, and you get ice. So those two are very, very close to one another. And so now the question is this hypothesis borne out by evidence? Is the EZ really necessary for ice formation? And I want to show you that it is. And so this is one of several experiments that we've done to demonstrate that. So you see a cooling plate down at the bottom and a strip of Naphion sitting there. And we put a droplet of water right next to the strip of Naphion. So everything gets colder and colder. And you see the region of view in the next slide. So we're getting colder and colder and starting to freeze. And what you see here is that here is the piece of Naphion, and here's the water drop. And this bright region is the first region to freeze. We always get freezing right at this interface. It begins. Now it begins to spread, and you can see it's beginning to spread in this direction. And you can see down here that it's spreading right in the exclusion zone before it actually goes out here. So it looks like the exclusion zone is the first to freeze. In other words, it is the exclusion zone that undergoes the change to ice. So the scheme is something like this, where you have an exclusion zone, and next to the exclusion zone are the protons, and they keep building up, you see here. And then what happens is, next step is they actually rush in to the exclusion zone, and as they rush in, remember you need protons plus exclusion zone to create ice. And so basically you have ice formation right here, and the process keeps repeating, and gradually you build up more ice from the exclusion zone plus protons. So that's the scheme. Now, does this really happen? And here you can see one experiment that we did to demonstrate. This is a chamber, and it's got some microspheres here, and we start cooling this cooling plate, and it turns out that next to the cooling plate we get a very big EZ here, and we put dye in, and the question is, if we put dye in instead of the microspheres, is it true? Do we see a pH change here indicating that there are protons that are rushing in? And you can see the result here. It shows that this zone that just freezes turns red. Red means a lot of protons in here, as I've shown you before. So it looks as though a lot of protons are rushing in during the time of freezing. Another experiment that shows the same thing. We have a cooling plate here, a little dish with water, and pH dye. So this means neutral pH. So we're cooling from here, and gradually this gets colder and colder, and it starts to freeze. It starts to freeze around the edges, and you can see the color around here has gone from the green to this red-orange, and the pH here corresponds to pH 3, huge number of protons. And the same with a droplet that's undergoing freezing. So here it's neutral, it gets colder and colder, and you can see the red color during freezing. So it's clear that ice formation absolutely involves protons. And so the ice formation is the exclusion zone, plus the protons give you ice. And so I think the energy paradox is actually resolved when you think of this process that's going on, because you're starting with an EZ, which is a liquid crystal, so it's moderately ordered, and the ice is more ordered. Now, so if you need more energy to get more order, you actually get the energy because the charge separation is potential energy. So you're adding this potential energy to get something that's more ordered. So there's no violation of any thermodynamic principle. Increased order does require energy. Now in the reverse case, if you need EZ water to get ice, then if you melt the ice, you should get EZ water. We tested that, and we got exactly that. Melting should produce EZ water. So in a little cuvette, we put some ice, and we gradually let it melt, and we looked at the water that was forming during the melt. And if you remember, EZ water is tested by looking at this 270 nanometer absorption. So here's what we found in the melting water, melting ice. There are 4 graphs, and each one is a different kind of water, deionized, distilled, et cetera, and you'll notice that they all have peaks at 270 nanometers. Ordinary water doesn't have that. Okay, so now, getting there, almost there, low friction. Why is there friction to start with? Well, friction is caused by asperities that, like mountaintops, and if you shift one by the other, you get friction. If you have 2 surfaces that look like this, a bit ragged at the edges, and they're hydrophilic surfaces, and you add water, so you add water. Now what happens when you add water? Well, you have an exclusion zone here, an exclusion zone here, and positive charges, hydronium ions here. The hydronium ions repel each other. They push the surfaces apart. Therefore, if you shear back and forth, you ought to get very little friction. And people have demonstrated exactly that with polymers, very little friction. Now, what about ice? What about ice skates? Well, it's been known since Michael Faraday that on top of ice there's a liquid layer. And you'd say, well, if you have a liquid layer, friction should be reduced. But I think it's more than that. It's possible that this layer that we're talking about contains EZ plus protons, because I just showed you that when ice melts, it gives you EZ, and together with EZ, you have protons. So you have a lot of protons sitting beneath that skate. And remember, those protons are repelling each other, so therefore the skate blade is repelled. It keeps away from the ice, and that's why the friction is as low as it is. You can imagine also that the more pressure you put on, the more protons you get. So you put pressure, you squeeze out those protons. You put more pressure, you squeeze out more protons. And the more protons you have repelling, the lower the friction. So the ice skater bears down really hard, and the friction goes down. Therefore, you have low friction. Biological implications, the last one. And so here's a question for you. How come when you do deep knee bends, you do this with almost no friction? Now I've got a lot of weight pressing down on my joints. So why is that? Well, if you look at the anatomy, the principle is actually very similar. You have a bone here and a bone here, and in between is a so-called joint capsule. And here you have cartilage, which is basically a gel. And here is cartilage, which is basically a gel. So next to the gel, you should have an exclusion zone here, an exclusion zone here, and protons in between. So you expect a lot of protons in between, and I just suggested to you what happens when you have the protons. They repel these surfaces, and people who are doing experiments on these joints report incredibly that people bear down, but they don't touch each other. The two surfaces remain some distance apart, and nobody can figure out why. And I'd like to suggest to you that the reason your joints don't squeak is because of those protons that are held in that joint capsule. Now, okay, so what happens now when you sprain your ankle? I don't know if you ever noticed, those of you who broke ankles, or so if you look at your leg or your elbow, I've been through it, the swelling occurs not in 30 minutes or so, it occurs in 30 seconds or less, 10 seconds. It immediately swells. So how does it immediately swell? Well, I think this has something to do with what we've been talking about. So the tissue that we're talking about in there is either connective tissue, muscle tissue, that's inside around those joints. Now, if you look, for example, at muscle tissue, so this is scanning electron micrograph of a muscle, and the muscle is running lengthwise this way, and you can see, these are mitochondria, by the way, you can see cross connections between these. Now, if you look at the fine structure of these myofibrils, we studied muscles for 30 years, it looks something like this. Here are the protein filaments, and notice something interesting. These protein filaments are all cross-connected. They're like rungs of a ladder. So the situation looks something like this. Now, these are hydrophilic proteins that run this way, and they're all cross-connected. Now, when you have a hydrophilic surface, you know what happens. The water layers start building up here. They start building here, the EZ layers, and they want to expand the structure. But they can't expand the structure because of these cross-links. And that's why your muscles or your tissues don't expand. They have only relatively little water in them. It's only 2/3 water. Other gels are 95% water. It's because of these cross-links that don't allow it to expand. However, if you have an injury and you tear the tissue, what happens is that these cross-links break. And if they break, then these EZ layers can pile up with impunity. They just build up. And I'd like to suggest to you that without that restraint to swelling, that's what happens when you sprain your ankle or you break your ankle. You get this, because the EZ layers build up within seconds or minutes. And so the swelling arises from that. Okay, finally, the last 3 minutes or so. Is that okay? Oh, I'm okay, then I can relax a little bit. Thanks. I was hurrying. Now I'd like to talk about calories taken in versus the amount of energy that gets used. See, it's not just the food that we eat, remember, that we get, from which we get energy. It's the radiant energy that comes in. We're full of water, right? And I'm receiving radiant energy all the time from outside, from the light here, that light, and the walls. And of course, plants use this energy to their advantage. And the question is, can we make use of any of this energy? Of this received light energy. If you take your hand and you shine a flashlight in the dark, you can see the light coming through. So some wavelengths do penetrate pretty far, and the question is whether we make use of that energy. And one possibility, a place where we might make use of it, is in the cardiovascular system, because the vessels are pretty superficial. Now, obviously, the heart is pumping and generating pressure in our arteries and whatever, but when you get down to the capillaries, you have a bit of a problem. So the red blood cells look like this, and they're typically 6 or 7 micrometers in diameter. Young people, the capillaries, the smallest vessels, are actually less than 6 or 7 micrometers. Some of them are as small as 3 or 4 micrometers. And so, you know, if you're a mechanical engineer, you're not going to design the system this way where you try to take something big and stuff it through. But that's actually what happens. And the question is whether there's enough energy from the heart to actually drive it through. And my Russian colleagues have done some calculations. I'm not sure if they're right. They calculate that if the heart really pumped the blood through those narrow, forced it through those narrow capillaries, the heart would need to develop 10 to the 6th times the pressure that it actually develops, because the resistance is so high in those vessels. So the question is whether maybe there's some other energy that helps that. So if you actually look at some vessels inside of muscle tissue, you can see that the red cells are distorted, they're bent, they have to squeeze through. They don't just sail right through like that, and some actually are retarded quite a lot in their passage. So the question is whether, you know, it might be that this gets some assist from the energy that's coming in from outside. Now that would seem ludicrous, a ludicrous idea if I hadn't shown you that we actually see it in the laboratory. We have a hollow tube, a hydrophilic tube, just like the vessels, sitting in water. And I've shown you that the radiant energy that comes in is driving the flow through vessels. So I raised the question, no answer, might radiant energy help drive your blood flow? Might we take the absorbed external radiant energy in assisting with that? So this is actually more general than just the cardiovascular system, you know, because in cells, we have, of course, the cells are much more crowded than I've shown here, but around each protein will be an EZ, negatively charged, and in the water that's beyond that, it'll be positively charged. So you have negative charge and positive charge separated from one another. And all the reactions that occur in the cell require either a negative charge or a positive charge. They're reduction or oxidation reactions. So what you have basically is a ready-made system to drive those reactions. And the energy for that can come from ambient radiant energy or heat energy generated by other regions in the body. In fact, all those protons that are generated is very similar to some of you who know biology about mitochondrial proton pumps. This is a proton pump, and you get it free in a very simple way. So separated charges drive reactions. So I think the exclusion zones are central for biological function. And just at the last moment I want to mention two things, is that if the EZs are central to biological function, now what wipes out biological function, at least temporarily? Well, anesthetics do, right? Anesthetics block all nerve conduction and many other aspects. And we did some experiments. I'm not going to show you the data, but I don't want to keep you too long. And we found that in clinical concentrations, the anesthetics, two local anesthetics, bupivacaine and lidocaine, diminish this zone and bring it to 0 in clinical concentrations. And in the reverse, aspirin, salicylic acid, which improves function. And so, you know, if you have a headache, it goes away. If you have soreness, it goes away. If you have fever, it goes away. You take aspirin, it's good for you. So we thought, well, you know, maybe aspirin actually increases the exclusion zone. And sure enough, that's exactly what we found. I'm not going to show you the data. It's not, the experiments are ongoing, but they show it. So I think that the EZ water is central to all biological function, which is what I suggested in that book. New book, as I said, is coming soon. And the last couple of slides, practical use of energy. So, you know, the sort of almost no-brainer is that if you have charge separation, that you ought to be able to capitalize that to get energy. And we've put electrodes in there, and the energy, and Kurt Kung, if you want to speak to him, he's here, is working on this project. It looks quite promising to get electrical energy out of this. And this is from the sun, energy from the sun using water, a renewable resource. And finally, obtaining drinking water. If you have a hydrophilic material and you put contaminated water next to it, that is water that's got junk in it, the bacteria, colloidal particles, whatever, all the stuff gets excluded. And the exclusion zone is pretty big, this EZ. And so if you could collect this EZ water, that should be free of contaminants. We don't know for sure, but we think that salt might be excluded too. And if salt is excluded, then it might be possible, we don't know this yet, to take ocean water and get drinking water from the ocean water. And this would be using the energy from the sun, not doing reverse osmosis where the energy requirements are huge. So the main conclusions, one is that the phases of water. So we know the three phases, and it looks as though the EZ phase is another phase of water. And if you take account of the fact that water has not three phases but four phases, then you can begin to understand many phenomena, including many everyday phenomena, a very small fraction of which I've been able to talk about. Another point is that a glass of water is not at equilibrium with the environment. It's a transducer that takes energy in and converts that to other kinds of energy. So where we've come is starting with the idea that the water is absorbing this kind of energy. I've suggested to you that this energy may be very important in various biological processes. We have no clue yet. This is just at the beginning to understand that water or the radiant energy absorbed in water might play a very critical role in all of biology. In chemistry, well, if you read the chemistry books, anything that's dissolved in water or suspended in water is just, it may have one ordered layer of water around it. And if what we presented is right, it means that that paradigm is totally different. It means that basically what's in the chemistry book in aqueous solution or suspension may need radical reinterpretation. In terms of weather, well, weather prediction is mostly based now on temperature and pressure. I think charge is much more important. And I think to get better weather predictions, if you take account of the charge in the clouds and the charge in the atmosphere, you have a much better chance to get a better prediction. In terms of health, we were talking to various people about various kinds of water that contain energy. And these kinds of water have been demonstrated anecdotally, but in some cases more than anecdotally, to be able to reverse pathologies of various sorts. And I think this is very promising for the future. In terms of food, if you want to store food, prepare it, freeze it, you have to understand what water does, what happens during evaporation or freezing. I think we have a better idea now. I haven't demonstrated in detail, but we've demonstrated actually quite nice filtration. You can remove the EZ and it's free of contaminants, and possibly desalination and getting energy from the water. So I leave you with that, and I think water has been really exciting to see, and I think we've discovered some stuff that has some potential for the future. Thank you.